Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Welcome to your channel, The Anatomy Canvas. In today's class, we are going to discuss the histology of the connective tissue. So, learning objective for today's class are to discuss the extracellular matrix of the connective tissue and different types of the cells which are present in connective tissue. Extracellular matrix, the ground substance of extracellular matrix basically consists of the three main components, that is the glycosaminoglycans, also called the GAGs, the proteoglycan and the multi-adhesive glycoproteins. So glycosaminoglycans are linear unbranched polysaccharides, for example heparan sulfate, chondroitin sulfate, keratan sulfate, hyaluronic acid and dermatan sulfate. They are very hydrophilic due to abundant negative charges, that is sulfate group which is present, and because of this reason they attract the water. So the large amount of the water molecules are bound to these glycosaminoglycans. Except for the hyaluronic acid, they are usually bound covalently to protein core as a part of the proteoglycan. The next are the proteoglycans. Proteoglycans are formed of the core protein plus the glycosaminoglycan side chains like bottle brush, and they bind to the cells and other proteins and the extracellular matrix. So I explain all of these things to you with the help of the diagram. First, see what are the multi-adhesive glycoproteins. These are the large molecules with the branch oligosaccharide chains. These are the proteins containing numerous binding sites for the cells which are present in extracellular matrix, and also for the integrins, which are the signaling molecules and other extracellular matrix components. Types of the multi-adhesive glycoprotein are the fibronectin and the laminin. Laminin is basically present in the cell membrane. It bounds to the type 4 collagen. It is part of the epithelia. And fibronectin is present in the connective tissue. So important for addition of the epithelial cell to basal lamina via transmembrane integrins receptors. So the GAGs, the glycosaminoglycans, are sulfated and non-sulfated. These sulfated are the chondroitin and keratan sulfate and attached to the multi-domain protein core to form the proteoglycan monomer, also called the aggrecan. So this is the proteoglycan monomer, and in which you can say, see in the center you can see the core protein is present, and to core protein are attached the GAGs. These proteoglycan monomer is attached to hyaluronic acid by means of the linker protein which is present at the junction of these two. So these green color fibers are basically the hyaluronic acid, and to which these bottle brush like structure is the proteoglycan monomer. These are the collagen fibers which are present over here, and these are the different connective tissue cells which are present over here. So this is another detailed diagram of the extracellular matrix. This is the proteoglycan monomer, in which you can see the linker the core protein is present in the center, to which attach the GAGs, and this green color is the hyaluronic acid, and to which it is attached to the proteoglycan monomer by means of the linker protein. So these are the hyaluronic acid. The thread-like structure is hyaluronic acid, and bottle brush-like structures are the proteoglycan monomers, also called the aggrecan. These are the collagen molecules which are present over here. Again, have a look at this diagram, in which you can see the proteoglycan monomer attached to the center. You can see this is the core, this is the hyaluronic acid, and this structure is attached to the collagen molecules. So in detail you can see this is the hyaluronic acid. In center you can see the core protein. On the sides you can see the GAGs molecules, and behind this the pink color is the collagen type 1 fiber. So this is basically the structure of the extracellular matrix proteins which are present. Now the cells which are present in the connective tissue, there are two types of the cell. One is fixed or permanent residents, and other are free or the transient resident. So out of the fixed, the fibroblast and the myofibroblast, they are most common, than the macrophages, the adipose or the fat cells, the mast cells, and undifferentiated mesenchymal cells. And out of the transient residents, they depend upon the condition, whether there is inflammation present or there is an immune response So they migrate from the blood to the connective tissue. That's why they are called the transient resident. So these are the lymphocytes and the plasma cells, the neutrophils, eosinophils, basophils, and the monocytes. These are the differential cells. The first is the undifferentiated mesenchymal cells, which is present in the connective tissue. It is the permanent resident. It can differentiate into chondroblast, into adipocytes, into fibroblast, into the mesenchymal cells, into endothelial cells, and into osteoblast. So these chondroblasts then differentiate into chondrocytes, and osteoblasts differentiate into osteocytes. Again, the hematopoietic stem cells, which can form the B lymphocytes and then the plasma cells, or they can differentiate into monocyte, the mast cells, the neutrophils, eosinophils, and the basophils, which are the WBCs, and also can differentiate into the red blood cells. The another type of the cell which is present over here is large cell. This is the this one is the megakaryocyte, and this monocyte differentiate into either the macrophages. It depends upon which type of the tissue is present. So in case of the bone, this is osteoclast. In case of the connective tissue, this tissue macrophage is called the histiocytes. So these are derived from the monocytes. So this diagram shows that all of the cells are derived from the undifferentiated mesenchymal cells, and then they differentiate into different types, which will form the different types of the cells. So first of all, we will discuss the fibroblast. The fibroblast is the principal cell of the connective tissue, and it is derived from the undifferentiated mesenchymal cell. Single fibroblast is capable of producing all of the extracellular matrix component. Most of the histologists describe the two types of the fibroblast into the either active form or the quiescent form. Most of the time these are present in a quiescent stage, but they have the capability to differentiate into active stage. So in these two diagrams, you can say this is the fibroblast, and in routine H&E stain section, it is only the nucleus of the fibroblast is seen, while the cytoplasmic processes are difficult to identify because they blend with the surrounding connective tissue and the fibers. Over here also you can see this is the active fibroblast, and in which you can see the strong basophilia as well as and large size of the nucleus. This one is also the active fibroblast. This is also the active fibroblast, while this one is the quiescent stage of the fibroblast. It is also called the fibrocyte as well. So fibroblast in quiescent stages, while in H&E preparation the nucleus is elongated or disc-like, as you can see in this slide, with the nucleolus most of the time present. The thin pale staining flattened cytoplasm that form bulk of the cytoplasm are usually not visible. As I have told you that their cytoplasmic processes are not visible because they blend with the surrounding extracellular matrix or the connective tissue fibers. They rarely undergo cell division unless tissue is injured, which activate the quiescent cell. Then it becomes the fibroblast. Initially it is called, in quiescent stage it is also called the fibrocyte. So they play a major role in process of the wound healing and response to an injury by proliferating and enhanced fiber formation. So active fibroblast, this is the electron micrographic picture of the active fibroblast. Here you can see the nucleus of the fibroblast cell, and in this area you can see the large number of the large profiles of the Golgi apparatus as well as the rough endoplasmic reticulum is present. On the sides you can see the cross section of the collagen fibers. So cytoplasm is more extensive and may display basophilia, and this basophilia is because of increased amount of the rough endoplasmic reticulum which is associated with the protein synthesis. And transmission electron microscopy shows fibroblast cytoplasm exhibit profiles of RER and prominent Golgi apparatus. As you have seen in this slide, these are the profiles of the Golgi apparatus and the rough endoplasmic reticulum. Then another type is the myofibroblast. These are not easily distinguished from fibroblast in ordinary H&E section, and with TEM the myofibroblast displays clinical characteristics of the fibroblast along with the characteristics of the smooth muscle cells. So that's why it is called myofibroblast, because it has some of the characteristics of fibroblast and other characteristics of the myofibroblast. So in addition to rough endoplasmic reticulum and Golgi apparatus, the myofibroblast contains bundles of longitudinally disposed actin filament and dense bodies similar to those observed in the smooth muscle cells. So these are the properties because of which it resembles the smooth muscle cell. That is why it is called the myofibroblast. The property, the point where it differs from the smooth muscle is that it lacks the surrounding basal lamina, while smooth muscle cells have the prominent surrounding basal lamina in the form of the external lamina which is present, but fibroblast lack this surrounding basal lamina. So these are the characteristics. Because of these myofibroblast resemble both the fibroblast as well as the myocytes. This is the slide of active and inactive fibroblast. In this area you can see these are the fibroblast nuclei and these are present in inactive stage, while here you can see the active fibroblast, and this active fibroblast in this H&E stain section shows strong basophilia. This is because of the large amount of the rough endoplasmic reticulum. This is another fibroblast. These are the other fibroblasts present in this section. Next is the adipocytes. As adipocytes predominate in adipose tissue, they are very active cells in many functions. Triglyceride storage and glucose metabolism is related to them. Secretion of the many bioactive molecules. Only two types of the adipose tissue is present, either the white adipose tissue or the brown. The white adipose tissue is unilocular and the brown adipose tissue is multilocular. Multilocular brown adipose tissue is usually present in the newborn baby in the first year of the life. So this one is the slide of the white adipose tissue, where you can see the unilocular adipocytes. All of the cytoplasm is filled with the fat droplets, and nucleus is pushed to the periphery in the form of the ring, and the cytoplasm is pushed in the form of the ring, and nucleus is pushed to one side, and it gives an appearance of the signet ring. So this is the slide of the white adipose tissue. Here you can see large single droplet which is present over here. Then the adipocytes, at the higher magnification you can see the large unilocular adipocytes which are present in the white adipose tissue. So this is the nucleus of the adipocyte which is pushed to one side, and this pink color is because of the cytoplasm which is pushed towards the periphery because of this large lipid droplet which is present. In addition to adipocyte, you can also see the blood capillaries which are present over here, and this is the electron micrographic picture. You can see the nucleus of the adipocyte, and this is the large fat droplet which is present, and cytoplasm which is present and pushed towards the periphery. Next is the brown adipose tissue or the multilocular. You can differentiate between these two. This one is white and this one is brown. This is unilocular and this one is the multilocular. It is present in newborns and also the hibernating mammals and involved in the thermoregulation. Upon naked eye appearance, these tissue appear red to tan in color because of two reasons. The one, that they are highly vascular, and second, they contain large amount of the mitochondria which contains cytochrome. That's why they appear reddish to tan. Third type of the cells are present in the connective tissue, or they are among the resident cells are the macrophages, and these macrophages are derived from the monocytes. Monocytes are present in the blood vessel. They escape from the blood vessels, enter into the connective tissue, and then they differentiate into macrophages. So you can see over here the arrows shows large number of the macrophages which are present. Their primary function is phagocytosis and antigen presentation. So macrophages are the phagocytic cell. Here you can see the large macrophages which are present, another macrophage and another macrophage, and in case of the connective tissue, these cells are given the name of the histiocytes. They are derived from the monocytes of the blood. So this is electron micrographic picture. As we know that these are derived from the monocytes, so that's why these cells contain large amount of the lysosomal granules. Usually the nucleus is a kidney shape or indented and can reveal the staining for the acid phosphatase activity with special dyes. In addition to these features, macrophages also exhibit large amount of the Golgi apparatus, the smooth and the rough endoplasmic reticulum, mitochondria, and these are all the secretory vesicles which are present in it. The surface of the active macrophage show many foldings. Here you can see the many foldings of this active macrophage showing you the phagocytized area, and these are the ingested vesicles which are present in it. The foreign body joint cell is another type of the tissue macrophage. So if a large foreign body is to be phagocytized, many cells combine together and they form the foreign body joint cells. So this is in case of the infection or the large virus or the some of the neoplastic cells which cannot be engulfed by the single macrophages. So the large number of the macrophages surround the foreign body and they become the foreign body joint cells. These are the ultrastructural features of the macrophage. Here you can see the nucleus of the macrophage along with the nucleolus. In this area, the dark color area shows the lysosomal vesicles which are present, and also you can see in this area, this one is the another lysosomal vesicles. In addition, you can see profiles of the Golgi apparatus and the rough endoplasmic reticulum which are present. Nucleus is usually indented. It resembles a kidney shape appearance. So these are the phagocytotic vesicles which are present. You can see these are phagocytotic vesicles, and these areas shows the secondary lysosome. This dark color area shows secondary lysosomes. Next is the plasma cells. The plasma cells are derived from the B lymphocyte and they constitutively secrete the antibodies. So here you can see the plasma cells which are present. Typically these plasma cells, the very important feature is first they have the secretory granules, and second their nucleus show typical cartwheel appearance because of the heterochromatin which is present, and this cartwheel appearance of the nucleus help us to identify the plasma cells in the light microscopy. This is electron microscopic picture of the plasma cell, in which you can see the nucleus. The area which are stained darker are the heterochromatin, and area which are stained lighter are euchromatin in this EM section. And these are the profiles of the endoplasmic, rough endoplasmic reticulum as well as the Golgi apparatus. Small amount of secretory vesicles are also present. Again, you can appreciate in this area the cartwheel appearance of the nucleus, the nucleolus which is also present. These are the Golgi apparatus, the mitochondria which are present, and large number of the rough endoplasmic reticulum. So plasma cells, the electron microscopic picture, and these cells are responsible for secretion of antibodies. Then is the mast cell. The principal function is the storage in secretory granules and regulated release or degranulation of the histamine and other vasoactive mediators of the inflammation. So because of these a large amount of the secretory granules, you can see these secretory granules in the light microscopy as well, and these cells show metachromasia, a very important feature, because when stained with tolvedine blue, the granules bind the dye and change their color to red. Usually they are darkly basophilic in nature, but because of the dye with the tolvedine blue, their color becomes the red. They are responsible for immediate hypersensitivity response, characteristics of allergies, asthma, and anaphylactic reaction. And these are the types of the transient cells which are present in the connective tissue. The connective tissue mast cells are found in the skin, in the peritoneal cavity. In the mucosal mast cells are in mucosa, digestive and the respiratory tract. So you can find the mast cells in different locations. This is the electron microscopic structure of the mast cell. Here you can see the nucleus of the mast cell, large number of the granules which are present, also occasional mitochondria present. This is the section of the elastic fiber which is present over here, and these are the section of the collagen fibers which are present in the connective tissue. So here I complete my 2 days class. The references are Basic Histology by Lekus and Siddiq, the Histology Text and Atlas by Jan Quera, and Histology Text and Atlas by H. Ras. Thank you very much for your attention, and Allah Hafiz till the next class.